I'd like to introduce Dr. Mirella Ortiz from Sao Paulo, Brazil, doing her undergraduate there, coming to Colorado State and doing her master's and PhD degrees. Currently, Mirella is serving as an assistant professor in weed science at Utah State University, focusing in weed physiology, molecular biology, and herbicide research. Her list of accomplishments is outstanding for this early in her career, having won just about every graduate student award she could win through Aquatic Plant Management Society, the Western Society of Weed Science, and numerous graduate student speaking contests. Dr. Ortiz is off to an outstanding start in her career. We are very fortunate to have her here to interview for this aquatic weed scientist position. With that, Dr. Ortiz, I will turn it over to you. Thank you.
Thank you so much again, Stephen. Thanks for whoever said on Zoom that you couldn't hear. That's appreciated. And I want to thank everyone for being here today. I know it's summer, it's a busy time, and it's not easy to make the time to go through all of these interviews. So I definitely appreciate you all being here today. So my research seminar, I have 45 minutes. I'll try to stay in time. I do tend to get a little overly excited and speak fast and go too long. So calm down and stay within time. So I'm going to talk about from the lab to lakes, optimizing aquatic weed management is just the title of my research, but I'm gonna actually usually needs one click. There we go, towards. So the line of my presentation today, gonna talk a little bit about my background, although Stephen did an amazing job, so I can save some time there. And then my research program, what I did at Colorado State University as a graduate student, what my program looks like at Utah State University right now, and then quickly share a vision for a program at UF if I'm offered this position, and my teaching philosophy. I know I have time for a teaching seminar this afternoon, but I only have 20 minutes, and I'm very excited about teaching, and I wanted to make sure I had time to cover that. So as Stephen said, I'm originally from Brazil, from the state of São Paulo, so southeast of Brazil. I'm from Ribeirão Preto, this town in red right here. It's in the countryside of the state of São Paulo. And although I didn't grow up on the farm, I would never claim that. I grew up going to the farm every weekend. So my family in Brazil, they have a sugarcane farm, and that's where I have my best memories growing up with my grandparents and just running and just eating some sugarcane straight from the field, which was amazing. So I knew I wanted to do agronomy for my whole career, pretty much my whole life. When people asked me what I wanted to be an agronomist. My dad's an agronomist, my brother's an agronomist. So I never really stood a chance. My whole family is connected to ag, and that's when I went to Piracicaba to get my bachelor's degree in agronomy. So here just quick about my education. So I got my bachelor's in agronomy. I thought I wanted to work in crop production my whole life, and I did internship in crop production, and it was not what I dreamed. It's like, that's not what I want. So then I did several internships, logistics, soil fertility, that went horribly wrong. And then I found the weed science group, and that's where I was happy, doing terrestrial weed management. So my bachelor's thesis is control volunteer Roundup Ready cotton, corn, and soybean, and the following Roundup Ready crop. And as part of my bachelor's degree, I had the opportunity to go to Colorado State University to do a mandatory internship with Dr. Scott Nissen. And that was the first time that I was introduced to the aquatics world. So although Brazil, 70% of our electricity comes from hydroelectric, we don't have any good aquatic weed management plan. Registering herbicides in Brazil in water is even harder and more expensive than here. We have 3 agencies overlooking everything, and if one of them says no, it's a no-go. So we don't really have any options. And then during my time at CSU as an RA, I had the chance to be exposed to so many different projects, invasive species, row crops, aquatics. So my background is really diversified, thankful to my advisor at the time. So then after doing a year and a half of RA at Colorado State University, I started my master's, focusing almost exclusively in aquatics. So my thesis title is endosoil behavior in 5 aquatic weeds. I was looking at this one herbicide and how it behaves in several different aquatic species, but still doing a little bit of terrestrial work on the side, because Colorado, It's not like Florida. There's also not a whole lot of water there, and my advisor could not fund a whole program on aquatics only. So I have a lot of that invasive species background and work too. I'll mention a little bit of the work that I've done. And then one of my committee members had an opportunity, a project also on aquatics. And when I finished my master's, he offered me a position as a PhD student from Dr. Frank Dayan. And my dissertation title is Xenobiotics Translocated in Aquatic Plants, a case study using 3 aquatic herbicides. So here just, I have some pictures showing everything I did at CSU. So worked in row crops, canola, soybeans, a lot of small grains, experience, specialty crops. So this is just planting onions. It was always an event. The onion bulbs were always late, and we always had the biggest crew and just happiest day planting onions on the field. A lot of transects, just mapping and analyzing invasive species. And of course, the aquatics program. Again, it was a small program at CSU. And I was at CSU for 8 years. Out of the 8 years, 6.5, I was the only graduate student working on aquatics over there. So I was in charge of over 20 tanks. Most of the research was done in greenhouse because of winter and everything. I do have field experience, but the majority of my background is focused on lab and greenhouse work. So the aquatic research program that I was kind of leading at CSU before I went to USU, I'll mention a little bit of the field projects that I had and the exposure to field aquatic weed management, and then greenhouse, and then going to the lab. So again, my background, Scott did an amazing job exposing me to all 3 areas of research within aquatics. So here, just normal field trials, just Scott and I, small boat, don't have anything as fancy as you guys have here, but we had a lot of fun on those boats. And this is specifically one project where we're looking at efficacy of 2,4-D and endothol combinations. And then a lot of that was just practical learning early on. So if you're out there in the field doing things, you learn like endothol and 2,4-D do not mix, not tank mix. And if you try, you end up with a huge mayo in your tank, and that's exactly what's in there. So that's how I learned. But if you're not sure, you do a small test first. So those are always fun experiences. Also, if you don't check the bottom of your lake for rocks and those kinds of things, you either lose your rake for analysis, or you go there and you grab it if you really need it. So this was my first year of master's, and just very thankful for all of that exposure and learning by my own experience. Did a lot of herbicide formulation trials, so just spraying either new liquid, granular, different formulations of herbicide, and that evaluations weeks after treatment. I had one chance of doing outdoor mesocosms, so that's the only outdoor mesocosms experience that I have. We had 20 tanks at the research farm at Colorado State University, moved everything there. It was about 20 miles away from campus, growing hydrilla plants. I needed a lot of plants, and I did just once because I could never grow. Hydrilla doesn't grow that well in Colorado, even in the greenhouse. So we don't have a hydrilla problem there. But I got everything. There was no water source nearby. Obviously, it was in the middle of the farm. So there was a canal running nearby. We filled all the tanks with canal water. And if you've ever done that, you know, the water would get algae very quickly. And there was no clean water source. And then this is 2 days after moving my plants, what the water looked like before starting the treatments. And just for 1 day, you can already barely see the plants. What an aquatic weed scientist would do, let's treat the water with copper. And that's what I did. And that's the color of my water tanks after doing the treatment. So if you have ever worked with copper before, you know, it's not supposed to look this blue slash green ever. You can barely see my plants in the tank. You can't see it. After I was done treating all the 20 tanks, I went back to the first one to see what it looks like. I was like, oof, that's not right. Got my colorimeter right away. And I learned that I added 10 times more copper than I should have added. And then I went to the label, I was like, okay, copper, hydrilla, guess what? Hydrilla is very sensitive to copper. Very few weed species are, hydrilla is one of them. So that's another early lesson, read the label, it's the law. I always say that to anyone that I start talking about chemical control. And double check your calculations. It doesn't matter how many times you've done that. Chances are that you still could mess up. So those are some of the good lessons that I learned early on during graduate school. The greenhouse experience that I have, this is a setup that I really liked at CSU, which is a very tiny miniature canals work. So we know there are very big differences between treating static water and flowing water. So with this small setup, we could simulate flowing water. So we would have the plants growing in flowing water, being treated as flowing water and kept in flowing water conditions, which does make a huge difference when you're comparing data with species that prefer to grow in flowing water. Though you can grow most of the species in static water as well, you don't get the same data that is translatable to the field when you don't simulate that water movement that the plants like and enjoy. A lot of just CT, the concentration exposure time experiments that any aquatic weed scientist will do, just different species, different herbicides. What are the herbicide requirement to control that weed species? I've worked with lots of species, different setups. I love the data analysis, native plants, so that's sago palmweed, flowering rush, amazing plant. I don't know if it's present here, but this plants I received on the mail from somebody, put in the cold room for 7 years, just in dry cooler. 7 years later, we put in water, pops up right away. So that was an experience. We didn't find anything that would control in our scenario really good. So it's frustrating, but so many more questions to it, the rest of aquatics. And then finally, going to the lab, I did a lot of herbicide physiology and weed physiology work, just seeing what happens with herbicide once it gets inside the plant. So that's one area that I love to work with. I did a lot of radio labeled work during my project, and I will present some of the data that was collected back then here today. So if you guys have any questions, also, I love to teach, and I do like to get back and talking, so feel free to interrupt me or ask any questions, or if you don't understand anything because of my accent, just let me know. So the majority of my master's was done with endothal. I did still a lot of work with endothal going through my PhD. That's what happens when you stay in one place for both of your degrees. My master's took me 2 years, and then my PhD was done in 3 years, which is nice when you kind of continue doing the same research. So although endothal is kind of an old chemistry, it was discovered in the 50s. We did not know the mode of action until 2010-ish. So it was almost 60 years for researchers to learn why exactly was endothal killing plants. So a lot of my work was done with that. It is a broad spectrum herbicide and a compact herbicide, and I did field greenhouse and lab trials, but I will focus on the lab trials today because I can't cover everything. So a lot of what I did during my master's was to look at how endothal behaves in aquatic plants. So what we did, we used C14 endothal, and we could track radiation inside the plant. So where is that radio-labeled endothal going inside the plant? Is it staying in the shoots? Is it going to the roots? We had a little wax layer at the top of that vial, so everything we found on the roots, we were sure got there by translocation and not by simply absorption. And then using a biological oxidizer or a HPLC connected to a radio detector, we were able to tell what was happening with that molecule inside the plant. So a lot of plant concentration factor numbers. So that's how we tell how much the herbicide is accumulating inside the plant. So we know the concentration of the herbicide inside the plant compared to the amount in the water. The only assumption we make when we calculate that number is that there is 1 ml of water per gram of fresh weight of biomass, which is not a bad assumption to make. Most of the submerged aquatic species, they're about 90% water content anyways. So that's not an expected number. And we do need to think about the log KLW of herbicide when we're looking at that number. So we know kind of what to expect to know if we should be surprised about our data or not. So the smaller the number, the more hydrophilic the herbicide is, and we would expect about the same concentration of herbicide inside the plant and in the water, so just reaching equilibrium. And the more lipophilic the herbicide is, the more it wants to go inside the plant, and we would expect higher concentrations of that herbicide inside the plant compared to the water column. So here, and again, I did this with over 10 species, don't have the time to show it all, and it gets a little repetitive. So here I have just the three first species that I worked with: Dioecious hydrilla, Monoecious hydrilla, and Eurasian water milfoil. We have endothel's log QLW right there, close to 2 at 1.91, so more on the lipophilic side of life. And we would expect it to accumulate in the plants somewhat, instead of just staying at that one number where it reaches equilibrium. So you can see that milfoil, we accumulate about 3 times more herbicide in the plant than in the water column. And then Monoecious and Dioecious hydrilla accumulated a lot more, closer to PCF of 7 for Monoecious and close to 12 for Dioecious hydrilla. The differences in the two hydrilla biotypes, it was somewhat surprising, but not so much. You know, it's like 2 completely different biotypes that will behave and respond different to herbicide treatments. Here's some desorption data. So what I also did a lot with different species and different herbicides is that once we expose this plant tissue to herbicide for, I can't remember if I did for 24 hours or 96 hours the exposure, then I would move that plant to clean water to see how much herbicide would come off the plant, because we know the concentration exposure time relationships are very important. The plant, your target species, needs to be exposed to the lethal dose of the herbicide for long enough to be controlled. If you have a scenario where that herbicide is dissolved or moved away because of flowing water, and your plant is not exposed to that herbicide anymore, how much can we lose after the plant absorbed that herbicide, and how fast would the herbicide come off the plant? So we see that Eurasian water milfoil, about 30% of the herbicide came off the plant 96 hours after being moved to clean water. So about 70% of the herbicide stayed inside the plant. And for Monoecious and Dioecious hydrilla was very similar, a little under 20% desorption. So majority of the plants stayed within that range. Very few was less than that, and very few was more than that. So we're retaining about 70 to 80% of pentothal inside the plant, which is not a bad number at all. And here's just some distribution. And remember, pentothal is a contact herbicide. So when we looked at some of this distribution, where the herbicide is going inside the plant, is it staying in the shoots or going to the roots? For some of the species, we were very surprised because we weren't expecting as much translocation to the roots of the plants at all. But here I have for 3 of them, and then a lot more. So I did, again, several species, several herbicides, just focusing on one herbicide here. Did native species. Oh, one graph's not showing. I did native species, invasive species, species not present in the US, Legercype, and so I'll talk a little bit about that project as well. So really fun to work with, and just to try to understand in general what was happening with endothol. And I graduated. I finished my master's, and then, but the question was still there, although my master's advisor was not my PhD advisor, he was still my committee. And then when you're working with radio labeled experiments, you can track the carbon-14, just the carbon-14. So we knew endothol was being translocated to the roots of aquatic plants, but in which form? Was it the parent molecule with herbicidal activity, or was it just one metabolite that does not matter? So that was the next question we asked, like, okay, in some species, we have 20% going to the roots. Does it matter? Is it a metabolite or is it actually endothol? So as part of my PhD, one chapter is looking at in which form endothol translocates to the roots of plants. So we used a mass spec to do that with cold, non-radio-labeled herbicide, and also radio-labeled herbicide to try to figure out exactly what compound in which form is getting where. We used 2,4-D as kind of our control. 2,4-D is a non-systemic herbicide, so we wanted to see exactly how much 2,4-D in which form was being translocated to the roots of aquatic plants. And let's look at endothol. So I used 4 ppm for both herbicides, the max label rate for 96 hours of exposure. And then I removed those plants from exposure, either to radio-labeled herbicide or not, extract all the herbicide from the plant tissue, and start looking at what form it was there. So here I have the data on the C14 and how much we could recover from the plant. And notice that I have 2 different plants here, milfoil and hydrilla. So for 2,4-D, our control-ish, if you will, we chose hybrid water milfoil, and for endothol, dioecious hydrilla. And although we understand that's not ideal, the 2 plants, they completely different species, they do not behave the same. The reason why we did that is because we know very little herbicide was going to the roots of the plants, and we just wanted to increase our chances of detection of the herbicide. So all of my master's experiments, I learned that hybrid water milfoil was the plant that translocated the most endothol to the roots, and dioecious hydrilla was the one that translocated the most endothol, or just endothol or 2,4-D? 2,4-D to the roots and endothol to the roots. So we're just maximizing our chances of detection, because although the mass spec that we used, sensitivity is amazing, we didn't need to worry about that too much. The radio detector sensitivity is not as good. So increasing that chances was just very favorable for us. Here I have the results on the radio labeled experiment. So I have at the top the standard injection and then extracted shoot tissue. So about at 14 minutes, we have the peak for 2,4-D standard parent herbicide molecule. So with radio labeled, this we cannot tell metabolites. We just know what is parent herbicide based on the standard. So we know everything that comes off here at 14 minutes. This is parent 2,4-D, and here about at 11 minutes, this is parent endothol. So we had a big peak for intact 2,4-D in the shoots of the plants, and one smaller peak here. It's a secondary metabolite. I did not go after figuring out what metabolite that was. And the same thing for endothol, one big peak of intact endothol on the roots of the plants, and a somewhat big peak as well for a metabolite that we don't know what it is. And there's no root data because we didn't get enough reductivity to the roots to detect. But that's when the LCMS comes in very handy. So here you have the mass spec, just the one graph on data showing the shoots and the roots. So when you're using a mass spec, you're able to tell the instrument the molecular weight of the molecule that you're looking at. So this is intact 2,4-D and intact endothol. In red, we have the shoots, in blue, the roots. So we see here right at little between 5.5 and 6 minutes, a big peak of 2,4-D for shoots and the roots. Again, 2,4-D is a systemic herbicide translocates to the roots. We have the peak for the roots there. And then surprisingly, we found about the same amount of intact endothol in the roots of aquatic plants as well. So although it is today registered as a contact herbicide, about 10% of the herbicide, the total herbicide extractable from plant tissue, was found intact in the root tissue of this plants. So they have similar implant behavior with 8 to 10% of the total absorbed herbicide going to the roots of plants. So a lot of this work is published in several different papers. There's still more to come. Done a lot of different work, but you can find all of that data online. Here is another project. Not going to talk about endothol too much anymore. On Fusilacor CET relationship in Lagersiphon. So what I love about this project, although very basic project in the weed and the aquatic weed science world, was working with a weed species that is not present in the US yet. So we were able to import Lagersiphon from New Zealand which at the time gave me a lot of anxiety. It's like, oh my God, what if like our side pick shows up in Colorado? It was me. But we were good. It was a contained greenhouse and everything. But just I love to focus in EDRR. Just prevention is the best method of control of any weed. If you avoid or if you have a good management plan to manage a weed before it shows up, that's the most economic way for you to do control. So this one we exposed Priscilla for 50 ppb for up to 1 week. And although you can see here in the picture that the treated shoots, they were damaged, it did not keep the plant from growing like crazy. So it's totally, these plants, if they were out in the wild, they would be able to start a new population. And it's also very surprising and important to keep in mind that Priscilla in New Zealand is very active in lagersiphon. So just different conditions, different water chemistries and temperatures and everything. Priscilla is an amazing herbicide. And then when we got the data, the company was like, wait, do you guys do something wrong? It's like, no, we repeated the same data and it's not an effective herbicide to control lagersiphon in Colorado, at least in our conditions, it wasn't. So you can't assume that everything that works in Florida will work in Colorado, Colorado will work here, or New Zealand will work in the US. So it's very important for us to address the needs of the state, but also keep in mind that we can expand and keep looking at potential other issues that we might have in the state. As I said, I did a lot of side projects. I want to mention just one because it's some different essays that I did, but Italian ryegrass resistant to PS1 herbicides. I had 2 populations resistant to PS1 only and PS1 and 2. And we were looking at the mechanism of resistance. What was causing this herbicide resistance in this species? So here just a quick picture of Italian ryegrass 48 hours after paraquat application of PS1 inhibitor herbicide. You have the susceptible plants, kind of hard to tell, but it's necrotic, chlorosis, so you're seeing that that plant's not going to do well. But when you look at the PS1 and 2 resistant plants, you see some damage, but the plants are a lot heavier than the susceptible one for sure. So we did a dose response experiment. In light pink, we have the susceptible, blue, the double resistant, and in purple, just the PS1 resistant plant. And there is our field dose right there. So you can see that at the field dose, all of our susceptible population, we achieved 100% of control of all of those plants. They are susceptible. But then for the 2 resistant populations, we did not, and the double resistant being even more resistant to just PS1 than the PS1 only resistant plant. So we know, actually, so then I did absorption and translocation. Obviously, I love doing radiation work. We wanted to see if absorption was reduced in those sensitive or resistant species. Translocation, we weren't expecting any paraquat to translocate. It is a contact herbicide. So we saw that there was really no difference whatsoever. All the plants were absorbing all of that herbicide. And we know by the mode of action that PS1 inhibitors, they usually kill the plant due to the ROS production, reactive oxygen species. So we decided to look at how much ROS was each of those plants producing. And this is what I have here, the ROS production. We can see that the susceptible population produced more reactive oxygen species than the 2 resistant because the plants were being controlled. So that was expected. And you see a decrease of that ROS production because the plants are dying. So they're not producing more ROS, the tissues are dying. So that was expected. And the 2 resistant populations, they're not producing as much ROS. Then we want to know why. Okay, so they're not producing ROS because the plants are not being killed. So everything makes sense. So we did a total antioxidant capacity. Are the plants not producing ROS because it can fight reactive oxygen species better than the other plants? And the answer is no. They have the same amount of total antioxidant capacity to fight those that ROS production. And I graduated. So then somebody else at CSU is now looking into that project, but we never learned the specific mode of mechanism of reaction of that population specifically to paraquat. So I graduated in December of 2022, went to Illinois, as Steve mentioned, for 4 calendar weeks. In January, one of the weeks being a huge snowstorm where campus was closed. So I was there working for 3 weeks, and I was offered the position at Utah State University, where I started in June of 2022. So I've been there for 2 years now. And I'll talk a little bit about what my program looks like over there. And I have aquatic with research in parentheses, because just like CSU, Utah is even worse. It's a desert, so there is not a lot of water. And although I have amazing support from my department head, the Utah Department of Ag, it is just, I cannot fund my program on aquatics only, so I'm still doing a lot of invasive species work. When I got there, I was blessed to get a huge lab space, and that's all I had, space. Nothing, I didn't have one single beaker in there. So over the last 2 years, I was able, between my startup equipment grants and research grants, to have a lab fully functioning, where you can do some cool research and work on. So I'm very, very proud of all of that, because it was not easy. Money goes so fast, and once you get into a factory position, you learn that. I remember as a graduate student, I'll just put an account, and purchase whatever I wanted, thinking that money went long ways and it was unlimited. It's not, it goes pretty quick. So I learned that the hard way. This is the greenhouse that I currently have, and that's all. I have 12 little tanks that I'm also very proud of. I currently have 6, as I said, weeds, but 6 aquatic plants growing: Eurasian and Northern, water milfoil, curly leaf, bird feather, monoecious hydrilla. Don't have dioecious. Bring some home. Just kidding. I wouldn't. Sago pondweed, and currently I have 2 funded grants to work on aquatics. One from UDEF, the Utah Department of Ag and Food, is a 4-year project, a little over 200,000. One project with the Department of Natural Resources, 1 year, that's actually ending at the end of this fiscal year. Right now, almost 20 grant. And I have a pending grant with the FR, the Foundation for Food and Ag Research, for a 3 years project, a little over 400,000. And this was actually an amazing opportunity that I was very happy to submit this grant a couple months ago. FR has a few grants that are for junior faculty only. So this one is the New Innovator Award grant, which is granted only for junior faculty that have received their PhD degrees within like 8 years of the application. Each land grant university can submit only 1 proposal. So USU did a screening, a lot of people submitted proposals, and mine was selected. So I'm very, that was already a huge achievement. I was very happy for that, but fingers crossed, because I could use that money to work on aquatics. And then as soon as I got to USU, I went out to try to figure out what aquatic issues Utah had. There's not a lot of water, but every single water body has a weed growing in it, which is very important. So I learned that the year before I started, a new population of parrot feather had shown up there. County weed managers, they have no idea what to do. Very scared of aquatic herbicides, putting herbicides into water. They don't want to do. So timing for me to start there was actually really good. We are, I sit on the state weed committee where we decide noxious weeds, what weeds are going to be listed, potentially delisted or moved categories. And we're just about to list hydrilla as in the watch group list, and milfoil for control. So weed managers, pretty soon they will be required by law to control their aquatic weeds. So that's one of the reasons why I have huge support from the Utah Department of Ag. And this one is just milfoil growing at Bear Lake. So here, just at this quick video for you to see. It's amazing to get to go to Bear Lake for work. So I won't say that it's not, but they just have, there's just a lot of milfoil growing there, and it's different than anything I had seen before. I call it the funky prostrate milfoil. So the plants, they do not try to reach the water surface flower. So they're there, population is growing, but the plants are never flowering, for most part of Bear Lake. Also, here's just the first application at Bear Lake that was part of with, Termacnet. It's the one doing all the applications at Bear Lake, so it was just, there's not a whole lot of water, so that's my joy. I get to go to Bear Lake to do research. So I submitted this project to the DNR, investigating herbicide efficacy and behavior for controlling water milfoil in Bear Lake. My PhD student Ola was the one working in this project. And so the problem is that we had 3 years of failed Prosilocor treatments at Bear Lake, and Prosilocor is a highly active herbicide in milfoil. So it was a huge surprise when we saw all this failed treatments in 19, 20, and 21. So the populations were not only not being controlled, but they were expanding at a very fast pace. In 2022, I did genotyping assay, so we had both invasive and native milfoil growing at Bear Lake, but no hybrids. Best guess, no hybrids, because plants are not flowering, except in some isolated dock areas. So here is just in 2022, the lake level was so low, and due to just proper water temperatures and everything, so here the plants, they do reach the water surface and they do flower. So potential for hybridization, those hybrids going to the lake and just making management a lot harder exists. So it was very important to kind of figure out what was going on with the plants. And this is what looks to do aquatic weed research in Utah. So you go to Bear Lake, you collect water and you go back to the greenhouse because of all the snow. We don't have plants growing there for 7, 8 months of the year, but in the months that we need to use the water body, they're growing. But then it's also a hard timing to do all the management we need to do, public perception using herbicides and all of that that you guys are used to. So just thought for you guys to appreciate Florida, where you can actually do the research. That's what I'm dealing with in Utah. So here's just some of Ola's data. He's done a lot of research. This is the ones that I chose to show. So he grew original milfoil in Bear Lake water and tap water for 3 months. So we can see the difference in the plants growing. Obviously, Bear Lake plants are a lot happier. I have the next slide, just the water quality and specs showing. So that was not unexpected. This is just the plant height. We can see in green Bear Lake, blue tap water. So plants are just growing faster, better, happier. Here number of lateral shoots that each plant produced in a period of 3 months. So we planted 1 10-centimeter shoot in each pot and let them grow for 3 months. So we had about 8 extra shoots in the Bear Lake water and only 3 in most of the tap water plants. And then we wanted to see if the herbicides were degrading faster in Bear Lake water. So why weren't the plants being controlled? They're not only growing faster, but is the herbicide half-life or degradation being faster in Bear Lake water? And the answer is no. So here we have Prosilocor. We also done for 2,4-D, but I don't have the data. But they half-life and degradation is about the same in both waters. So our best guess is that plants, they are just growing faster and they can't deal and handle and metabolize all of that herbicide a lot faster due to the faster pace. So herbicides are fine, the plants are just the super weeds. Here just some data. So the water quality of Bear Lake and tap water that we're using. Here is just the dry biomass of the plants that were growing for 3 months in the greenhouse. And this is treated plants. So we treated with 2,4-D at 1 ppm for 24 hours. All the tap water, we had about 90% of control, 95% of control. All of this biomass, 0.5 was just dead brown biomass. And then the Bear Lake plants, 1.27, and we had about 40% of control in greenhouse conditions. So the plants are definitely doing better and just growing faster. Don't know exactly what's happening inside the plants, but that's something we want to tackle, but just growing faster, they're just able to handle that herbicide a lot faster. Some of the additional research I'm doing Transiella. She's another of my PhD student, and she is looking at the potential volatility of a clonifene. So the issue that the company came for us was that there's a lot of off-target crop volatility injury from this herbicide, and there are some interactions of soil moisture and temperature. So Fran is in charge of this project. We sprayed glass lids just to see how much herbicide we would lose, used HPLC with a diode array detector connected to it, and used oxyfluorophane as our control group. Oxyfluorophane is a very volatile herbicide with a molecule that's very similar to a clonifene. So in the first 48 hours, we lost about 97% of the clonifene sprayed to the little glass lids. And a clonifene 48 hours after treatment, we didn't lose anything. So this was a good surprise. The company was very excited about it. So now we have more questions. We're gonna now add soil and sand and moisture to this scenario. And if any of you have worked with or read about EPTEN, EPTC, it's very similar. So there is like when the soil is at full water capacity, we lose more herbicide to a little bit of co-distillation volatility, just terminology. So then we lose more EPTC when the soil is at full capacity. So that's the next step for France projects. And the last side project I want to mention, I have time, is the influence of rainfall and the activity of S-metolachlor applied to wheat residue. So Fernando is a graduate student in Brazil. I was his co-advisor. I have signed a mock memorandum of cooperation with a university in Brazil, and Fernando was able to come to my lab for a whole month to do this part of his project. So just seeing how much herbicide we lose due to thatch on the ground. So I won't get into the data, it's just too much, but we had 3 tons per acre of wheat residue in little baskets. We sprayed both S-metolachlor alone and a pre-mix of S-metolachlor with fomazepam. I had 4 different rainfall intervals, 0, 4, 7, and 10 days, and 3 different rainfall volume. And the data is just amazing. So when you have rainfall the day of your application, we were able to recover about 90% of the herbicide. If you wait even just 4 days, then we lose about 75% of the herbicide. So the losses are just amazing, very important for farmers to spray right before the rain event. So vision for my program here, if I was offered this position, of course, all the big grants, USDA, NIFA, AFRI, in collaboration with a agronomy faculty in the department, Florida Fish and Wildlife Conservation, Southern IPM Center, internal grants, and today I learned about a lot more opportunities. I was Googling this one and I was a little disappointed because there was no plants in the last cycle. It's like, what's happening? It was just med school. But that was just the one time they told me, and I hope so. Research groups and societies and industry money, but ultimately coming to a program that's already established, you learn so much from who's already there. So when I interviewed for USC, it was the same thing. This is all my plans. When I got there, there's like all the other monies that you're not aware of and you don't know. And that's the best way for you to go. Just make those connections and learning from people that are there, what the best opportunities are. I would love to do a more basic greenhouse lab research focusing on herbicide physiology and aquatic plants physiology and biology. That's where I love. But again, my background is wide enough that I could do anything on the aquatics words. And ultimately what I want is address the needs of Florida and UF. So if there is the need of more greenhouse and lab work, that would be amazing. But if you're in a place where your research questions, although still important, they're not addressing the needs of the state you are, that's just a little hard to explain. You know, that we all know about the international recognized programs and this and that, and working with Legrosiphon from New Zealand, very important, but ultimately stakeholders, they want to know how to fix the problems that are here. So my goal is to find my niche and how I can contribute to University of Florida and the center. Some program goals, I'll cover this too much, I'm out of time already, but teach and I help understand and contribute to the Weed Science Certificate, establish connections with the stakeholders, the center, the agronomy department, pursue funds, and I have recruit students and apprentices because hopefully I'll have funding and desire for my students to come because they're amazing. Very happy with them. And then in 5 years, just have my own established program led in collaboration with everybody else in the center. But just have a very diverse program working everywhere, addressing the questions and needs of state of Florida. And just my teaching, currently teach fundamentals of integrated weed management. It's a cross-listed undergrad, graduate student level class. 4 credits, I have about 35 students each year. And I developed an aquatics lab. And the only thing I want to say about teaching is that we need to, with time, figure out what happens. So I developed an aquatics lab in 2022, did not offered in 2023, and I always ask my students, what you like, what you not like, and what should I change? And in 2023, 5 students out of 35 thought it was the best lab ever. And then, you know, this is important because you need to change. And for example, the monitoring method, I change, we increase the favorite, and sometimes you make changes that doesn't work, and that's fine, and you change it again. With seed dispersal, 9 people hated it, I changed, they hated it even more. Now 11 people least favorite, but that's the only way that we can to forward and provide the best to our students. You know, we see what's not working, we do different. Does this new way work best or worse? So I need to go in the other direction with the weed seed dispersal. But I was very happy to see that a lot of them appreciated the aquatics lab. I don't have time to this, but I'm just a student focused instructor. I love being in the classroom way more than I thought I would. And it's just amazing. I love the interaction with the students. They're just great. And that's what I had for today. Thank you for your time, for being here. If you have any questions, I'm here.
We got a few moments for questions here before we move on to the faculty discussion.
I started 5 minutes late, so I'm right on time.
All right, any questions from the audience first? Patrick.
Yes, thank you for your presentation. What was your favorite product you worked on, and why was it your favorite?
That's a great question. I think my favorite project overall, so I have 2 favorite from CSU and USU. CSU doing all the radio label work. I think I had so much fun, and I did way more than I was expected and required to do, because I was loved to see that. And actually I didn't have time to show, but what I did a lot was also combination of herbicides, you know, to delay the development of herbicide resistance, either rotation or combination. Combination being the one that delays the most the development of resistance. I did a lot of herbicide combination and keeping track, one herbicide alone, what does it do? The other herbicide alone, what does it do? And then radio labeling one herbicide or the other in combination. And it's amazing how much one can affect the translocation of the other herbicide or even absorption. So it's kind of, especially when you're publishing, it's kind of hard to talk about synergism and antagonism, you know, because that comes with just a heavy weight in words, but you see a huge impact on the herbicide behavior inside plants when they're combined. Would that have any, I guess, end user impact? Not necessarily, because we know in real world, the roots of the plants are also somewhat exposed to the plant. So in all of the lab research, we do have that wax layer. So we see how much of the herbicide is being translocated. In real life, the plants are just there. The soil is also exposed to the herbicide. So the roots could absorb that herbicide as well. So it doesn't necessarily mean that that impact would affect management. But it's very cool to see. And then for my time at USU, just this Bear Lake project is just, and many of you might have seen milfoil growing like that, but I've never seen it at Colorado. The plants were always growing up straight and flowering and breaking off and trying to start the new colony, the new population. And at Bear Lake doesn't do that. It's funny how you can see all of the plants growing, and you can see each individual plant, just there's like all the sand around, and they're not taking over the entire bottom of the lake, and it's such a puzzle to me, and I'm so happy that I'm able to tackle that and trying to figure out why are the plants still expanding but not thriving. Plants that are thriving, they're flowering, they want to go through the whole cycle. They're expanding, so we're seeing failed treatments, population growing, and still they're kind of just there. They're not plants that are thriving and flowering, but they're expanding, just like would love to understand why that's happening.
Thank you.
One question from Zoom, and I think the gist of it is really, why do you want to come to UF?
Why do I want to come to UF? Excellent question. I have several reasons, personal and professional reasons. Although I'd like to think I have a successful program at Utah State with a lot of support. It's not like Florida. Yes, I have 12 tanks. So having colleagues that I can discuss and collaborate with and talk about aquatic weed science. I'm the only aquatic weed scientist at Utah. And sometimes it's just hard, you coming up with all the ideas and troubleshooting and the next what to do. It's hard. So having colleagues would be amazing. There's no place like the center to do research. And personally, I'm originally from Brazil, so I can handle with hot and humid a lot better than I can do with 7 months of snow. It's a lot closer to Brazil, easier to get to Brazil. My husband's family is in Atlanta, which is a lot closer, so just a lot more family proximity. And let's just say that my husband and I, we didn't adapt to Utah as well as we thought we would. So just some of those difficulties personally and professionally over there. Thank you.
Let's see, Luke Flory. Luke, you have a question?
Oh, they weren't seeing me for most part.
Hi. Yeah, I wonder if for those of us that don't work in weed science, I wonder if you could take a step back and describe maybe two of your major research themes in general terms and why they are important.
Okay, sorry about that. The jargon and chemical control. So understanding all of this chemical control opportunities that we have with aquatic plants is very important because it is the most cost-effective way to control aquatic weeds, is using chemical control. And if we know what the best herbicide to control the weed in combination, as I was telling before, then we can delay that resistance development, so weeds that are not being controlled by a herbicide anymore. Because when you think about the aquatic world, although chemical control is still the most effective way to control the plants, we don't have nearly as many options as terrestrial weeds. So there's like over 300 herbicides registered for terrestrial control and about 16 for aquatic weeds. So we already lost fluridone to control hydrilla. If we keep losing herbicides to control those plants, you're going to be in very bad shape. And resistance development is a lot slower in aquatics than it is in terrestrial. We have herbicides being used in the same species for 20 years and resistance wasn't developed yet. But it's a numbers game. So there is one plant, one biotype that is out there that's resistant to every single herbicide. It's just a matter, have we sprayed that plant yet or not? So it's just for some reason that number games is a little better for us to play with in aquatics than terrestrial weeds. But understanding our chemical options and how to delay herbicide resistance will just provide us with the best long-term management and viability of those herbicide options going in the future.
Okay, last question here.
All right, Stephen mentioned during the introduction that you have some experience with molecular biology. I assume this was in regard to resistance mechanisms. Can you comment a little bit on that?
Yeah, so a little bit. I love doing molecular biology, but I learned during grad school that if you're not, you're either in or out. But I like to step in it. I like to do a little bit, but I'm not a molecular biologist. So a lot of my experience with molecular biology is doing genotyping assays, so things like PCR. So at CSU or USU right now, my lab provides a service to the Utah Department of Ag for genotyping milfoil, so every single lake in Utah has milfoil, either the native or the invasive, so my lab are in charge of telling, so they send a sample and we analyze all the samples. And also on the terrestrial side, we do a lot of gene amplification for glyphosate resistance, mainly Palmer amaranth and kochia. So my lab's providing that service, just analyzing and figuring out the mechanism of resistance to the Utah Department of Ag. So on top, as part of the herbicide physiology work that I love, there's a lot with mechanism of resistance. So just understanding how the plants become resistant to weeds. So at CSU, I had a lot more opportunities to work with that. There is a molecular weed scientist at CSU that I had a chance to work with and do a lot more research and trying to establish a little bit of that molecular capabilities in my lab, starting with providing the service where I can get money to buy the instruments, just trying to get more into mechanism of resistance for terrestrial plants, because aquatics, again, there's just one, and it's a lot slower process than in terrestrial, but I really appreciate that mechanism of resistance work.
All right, thank you. Okay, Mirella, I'd like to thank you again.